I will be presenting some general slides using a presentation to give you a gross overview on what ARIVIS is all about. And then my colleague David, who's sitting in the back, and I are going to switch. And David is going to give you a live demo presentation. Okay, so basically ARIVIS has been in the field for more than 10 years. And we have developed several software solutions which you will find in the typical imaging workflow. Basically, as soon as you have the digitized image and you want to do something with it. So you want to look at the data, that's the first thing. It sounds ridiculous, but I'm pretty sure that Mike can tell you some stories about looking at some of the data files which are generated using the clearing techniques, using the light sheet microscopy technique. So basically you will find our solutions from what we call transformation, and we will be giving you some examples here. Visualization, creating movies, animated movies, making screenshots to share it with colleagues, as well as doing analysis, whether this is manual or whether this is by automated image analysis. So you see here the logos of our 3 software lines, software products, which are combined as a platform. And they are based on our, what we call ARIVIS core technology. When we do the hands-on sessions, we will give you much more information about that, because this is one of the crucial concepts of our software. Having a dedicated file format, a dedicated file which the software will access in a very fast and robust way. So there's many concepts, or several concepts, which make us known to be the experts in handling large image data in the field, some of which we will be able to demo here right now. Which allows researchers in various fields. So now we are here at the Institute for Stroke and Dementia, but we have customers in other research communities using other microscopy techniques who can make use of our solutions, right? So this is just a, let's say, an overview on what imaging modalities would you be using or would you have used and generated data, and would then maybe do a follow-up or follow analysis in ARIVIS, and what kind of quantitative answers would you want to get out of this data. So as biologists, as researchers, you're often interested in interactions of structures or cells and other cells, or certain biomarkers within a certain region of the tissue and so forth. And we believe that with ARIVIS we provide a good tool or a stock of tools to get answers to that. We do so, and here I'm already jumping to our major desktop solution called ARIVIS Vision 4D. We do so by providing a software which would run also on standard PCs, which allows you to handle even a terabyte of data, and which would always be interactive, setting up, for instance, analysis routines, or would be interactive in creating animated movies. And you would really always be able to get a preview of what you do. And that is important, because if you have 500 gigabytes of data, you are setting up a pipeline, or you're setting up a animated movie, and you start to run, it runs overnight or is finished the next day, and you find out not so nice, I have to redo it. That's a pure waste of time. So basically, even though that might not be in your first thought when you think about software tools to aid in the imaging pipeline, to provide an interactive preview at all steps, or being performed at all steps, it will be vital as soon as you start light sheet microscopy. It might not be of relevance on small confocal subsets, but here it's really crucial for your workflow. So basically, if we look at ARIVIS Vision 4D, and we do so also in a live demo, what I've just said to wrap this up, so you will of course use such a software maybe dedicated to the large imaging workflow, if you use the Zeiss Z1 or the LaVision Ultra microscope or any custom built SPIM. But you can also apply, of course, the same software tool to other questions and modalities. And these are now screenshots and an animated movie created in Vision 4D, simply showing you the bandwidth of modalities. You see here, for instance, a very nice Drosophila with a color mapping, what we call a color mapping. So false colors are applied to give you a better perception of depth and of this specimen. So basically, if it comes to your daily routine work, it will start with the import of the data, and then you would probably, sometimes you may have to put single volumes together, which we call stitching, or a volume fusion even, and then you would apply nice color mappings, create movies. So basically, what I would like to do right now, just to switch briefly to our software. So here I have opened a empty window, and what I simply do is I'm using an ARIVIS file. I drag it into into our software, and it's opening this file from my external hard disk. So let me just see. And I've opened the file. There's a speciality also in our concept to allow you to work with different statuses of a document. So here I'm switching from the original file format, which consists of almost 90,000 planes, and I'm switching to the so-called stitched status of this document, which would be the result after making a data transformation using our tile sorter and our volume fusion. And what I'm just doing, and I will switch to the viewer of the software, and it is now creating a 3D rendering in this window. And you can see it's already loading the data. It is doing so tile by tile right now. But at the end, in my setup, which is absolutely not optimal, because I'm using an external drive. Everyone here with experience would always say never do that. And you would not work on a notebook in your daily life, but I can work with this data, and this data set itself is 800 gigabyte in size. So usually this is impossible on a notebook like this. No, commercial solutions use different strategies, and ours is to free the user from running the software on large RAM, large memory consuming workstations. Saying this, I can now work. I could work with this data. I could start working, making a movie, making a nice animation, as we have seen in Ali's presentation before. I can preview what I want, and you see, I'm of course setting up now something. I would do this in a nice choreography, obviously, and then I could export this movie easily. So that was just a short excursion into the software. David is going to give you a much better insight in a second. Simply kind of, if I'm always claiming until now that we are experts in large image data, I think I have to prove at some point. So that was the point. I've proven it for once. And now I would like to continue to give you a little bit more of a summary. So basically, what you would do is sometimes you would apply filters for visualization, for nicer visualization, which my customer did here on the left-hand side. And of course, like in Ali's lab, you can use the software then to really look at several image layers and to create screenshots as well as animated movies. But of course, you can also alter the data, resample it, crop it, export it to make it accessible to other software solutions, right? So basically, the visualization includes also, to a certain extent, to provide you with a tool to manage the future, basically, because the future, you are now starting with the clearing, or you may have started, and your future will be that you end up with 500 gigabyte, 1 terabyte of data sets. And at some point you have to look at the data, and at some point you want to share it with your colleagues, and you do so by following this particular workflow. So this is a nice movie from Hiroki Eda's group. Ali has mentioned it before, which we asked as a proof of concept a year ago, as we did not have access to that large files yet. So now our customers are generating many terabytes of data. At that time, the Ueda group was kind enough to provide us with a large set of small pieces of volumes of then altogether 14 terabyte, shipped it from Japan to Rostock, where our engineers are located. Did what Mike is doing, like every 3 days, I would think. You stitch data, you fuse data, combine it to a large volume when you have the whole mouse. And we did that for this data set, and as it is at a high resolution on a whole brain, the whole data set itself is then 14 terabyte, which is incredible, really incredible if you think about the numbers. So for you, in German we say, erstmal kleine Brötchen backen, so you will start small, right? So I'm also consensus with Ali. I think you will not start with a whole mouse clearing. You will probably start with the single organ clearing, and that is just fine. The software should still handle it if it's not 1 terabyte of data, obviously. But the principles should be fine, or I would like you to be reassured that you can use Vision 4D in this case really to handle whatever comes in the future. So basically there's functions like undoing and bookmarking, and I think this is all topics for the hands-on sessions during the workshop. So one last thing, if you look at, into the workflow, is the quantification. Usually as image analysts, you would always distinguish between manual editing, so creating your manual regions of interest, or even segmenting structures manually, or automated image analysis. And we have tools for this. We call this the ARIVIS pipeline, which allows you to make a scalable image analysis quantification. And what kind of numbers would you get out? So I have included this slide very generic, as our ARIVIS approach is rather generic. So You will have access to operators in a rather easy to use user interface in a stringent pipeline. And you can ask, for instance, where are the red cells, how many red cells, what is the volume of the total of red cells, and so forth. But you can be more sophisticated with this tool, asking, are the red cells in the vicinity of a vessel, or where are they located in the region of interest, and so forth. So basically, you can get out single cell information and information of groups. You can relate groups of objects or groups of cells or groups of spots to each other. One last thing, and this might be, so who is doing live cell imaging in general? Not here, but okay. So for the two of you, tracking is another extension, I should say, of the image analysis or quantification. So often, if you are doing tracking, you have problems with your specimen moving. So there are tools in ARIVIS Vision 4D which allow you to correct for this drift, and you can batch process the segmentation as mentioned. You can combine the object colocalization with the tracking, and at the end you can also edit created tracks if they are wrong. And the if I have learned is not often, but it is something in tracking. It's not 100% for sure, so you will appreciate that there's tools to edit the results afterwards. So coming now from quantitative analysis or automated analysis to the more manual approach. I've called it like where automation is not feasible or wanted, and this is a nice example as this combines two things. It is what Ali referred to as sometimes you want to look at normal versus abnormal, or in this case it's a tumor model. And you may not, for instance, if you wanted to depict the regions of interest here, the tumor inside, it is probably feasible to do this in an automated mode, but if you have to be precise, you'd rather do this by hand. And so there are tools, of course, in Vision 4D to allow you to do this. And here's some screenshots depicted from a workflow David and I did recently. And it is nice because at the end you can create numbers out of this, and you have, let's say, sometimes there is simply no other way than doing things manual, and you have very interactive tools here with preview again to do so. Whereas basically this leads already to, or if there are questions to Vision 4D, I'm happy to answer. Otherwise, I will continue. Yeah? Mm-hmm.
I haven't found yet if they try to get for a mean pixel.
The mean intensity of each pixel, but it is there already. So.
Just how can I extract it?
Mm-hmm. So we will show you later in the software, I think. But and I would be very curious why you would want to do this. So I guess you would want to do that in a certain region for a certain staining. So let's show you that later.
When you have your measurements ready, I think there's also a way to export this
Mm-hmm.
into a spreadsheet.
Yeah, but I think I should first understand what the reasoning is about, because you wouldn't be doing that for a complete, let's say, complete volume. You don't want to do that, but exactly, yeah. It's available, yeah. Yeah, even if you mouse over, you get the intensities, but yeah. We will go into that later. So I've shown you some example workflow steps if you were using ARIVIS Vision 4D, and it is really a routine tool. You can apply it to your routine work with the digital image data. And what we believe is being experts really in software development and software engineering. You can extend the functions which I've shown for Vision 4D in terms of exploration, going inside the data, going inside the microscopic image. And that is what we call the ARIVIS MUR. It's now the 3rd year then of development. I always have to think. And here we see a nice move. And we thought about having you here as a model for our demo. So we have this data set. You saw it before, but the color mapping was green. So in Ali's talk. So this is a nice screenshot of the brain data set, and Mike obviously can give details of the biology behind it. And it's a brilliant screenshot, and if you look at it in a 2D screen, it will also look lovely, but you're still blocked by the screen, basically. You're still flat in your perception. And that's why, like 3 years ago, ARIVIS started to really develop a individual application for using our core technology again, and applying special rendering techniques to show a microscopic image in the virtual reality world. So that you really immerse. And this guy here, this is a model, is wearing the Oculus Rift goggles, because those were the first really to be technical advanced at that time. And that is a data set, also a brain data set with a nice color mapping, and everyone who is of course invited to come to us during the next days and have this goggles or this experience himself. But before David, when I can show you some movies here, but David is going to give you the demo live, and it is a little bit more impressive. So we are going to screencast that here on this screen, and you can follow what David is doing. I think with this brain data set, or with the one from, okay. So basically, just before you do so, the advantage of exploration in VR was also first hidden to us, really. So we started and we thought, maybe just a marketing gimmick, and it is a cool marketing tool, I should say so, but it has shown to us by reaction of scientists who are looking at their own data set, and that is vital, that you can gain much more insight sometimes in the position orientation of a structure, or like here in this red skull, the orientation of the muscle fibers is important to my customers. So you are not blocked by any anatomy. Usually you would have to crop stuff away, and your brain has to rethink if you look at 3D data on a 2D screen, and this is simply, it's vanished. So you're inside the data, and with some, with the tools, you're going to see that, you can really navigate inside the data. So basically, there's an extension to that, and this is probably more reasoning also to you. You can do measurements in the VR, and you can do so very precise. You can nowadays also annotate regions of interest, and you can create de novo segments. So you're going to see that live. Basically, I've mentioned that we are providing a platform. I'm skipping this now, and this platform we are demoing here is Vision 4D and MVR. We have a web-based tool called WebView, which I'm also happy to give you some details later. What is important for you to know is that you can always ask for a software trial. Obviously, you can always ask us to demo on your own data, so you have our contact details, and basically I'm happy and I'm looking forward to the next days.